You know, usually when we talk about getting a medical diagnosis, there is this comforting expectation of precision, right?
Right, yeah, like engineering.
Exactly. Like if you break your arms, the x-ray shows that jagged white line, and the doctor just points at it and says, there it is, that's the problem. But what if I told you that recent large-scale data shows people who get the shingles vaccine, which is a shot for a very painful skin rash caused by a virus, actually have a significantly lower incidence of dementia later in life?
It sounds completely counterintuitive when you first hear it. I mean, it immediately raises this massive, unsettling question of, what on earth does a viral skin infection or the vaccine for it have to do with Alzheimer's disease?
And that bizarre medical mystery is our mission for today. We are doing a deep dive into the source material of a groundbreaking 2026 paper published in the journal Cell by Tyagi and colleagues.
Yeah, and the mission here is to uncover the exact molecular mechanism of how Alzheimer's disease actually spreads through the brain.
Right, because we're looking at it not just as a slow, inevitable decay, but almost like an infectious virus that is, actively using our own memory-building machinery against us.
It really is a fascinating shift in perspective. To get to the bottom of this, we're examining a single, incredibly dense piece of literature titled ARC Mediates Intercellular Tau Transmission via Extracellular Vesicles.
Quite a mouthful.
Oh, definitely. But basically, they mapped out a microscopic transport system. We're going to break down the exact mechanisms they discovered. And more importantly, the incredibly clever experiments they designed to prove that this is happening right now inside our heads.
So to keep us anchored as we get into the heavy biology, I find it really helpful to think of the brain as a crowded neighborhood. In Alzheimer's disease, toxic trash is accumulating inside the houses, which are our neurons, right?
I like that, yeah.
Today, we are going to talk about the exact garbage truck that moves this toxic trash from one house to another, eventually spreading the mess across the entire neighborhood.
Right.
And the main protein we're looking at today is called ARC. So for you listening, to remember what it does, just think of this mnemonic armored retrotransposon courier, ARC.
That's a great visualization. But to truly understand how this microscopic garbage truck works, we first need to define what the trash actually is and, why the cell wants to get rid of it so desperately in the first place.
Right. What's building up in the house.
Exactly. So the culprit here is a very specific protein called tau, T-A-U. Now, normally tau is an essential good guy. It stabilizes microtubules inside your brain cells.
And you can think of microtubules as the structural girders or maybe like the internal train tracks of a cell.
Right.
They allow nutrients to move up and down the long branches of the neurons.
Spot on. But in Alzheimer's, and actually a whole family of similar diseases we call tauopathies, like frontotemporal dementia, things go fundamentally wrong with that tau protein.
It changes shape, doesn't it?
It does. It undergoes a chemical change where too many phosphate groups get slapped onto it.
Okay, so it gets hyperphosphorylated.
Exactly. And in biology, adding a phosphate group is usually just a way to turn a protein on or off, changing its shape slightly. But when tau gets overloaded with them, it gets heavy, negatively charged, and it literally loses its grip on those microtubule train tracks.
So the tracks collapse, and this modified tau falls off and just starts clumping together inside the cell. These are the famous neurofibrillary tangles, right? The ones you always hear about in Alzheimer's pathology.
Well, that's the one. But the truly scary part isn't just that they clump, it's that they undergo a process called seeding.
Right. I always picture this like a zombie movie.
Yeah, that's exactly what it is.
Like when one of these misfolded toxic tau proteins bumps into a healthy normal tau protein, it corrupts it. It forces the healthy protein to misfold and basically join the zombie horde.
And that zombie-like spread is ultimately what destroys the neural networks. The original cell just fills up with these corrupt proteins, so that's our trash. Now, let's introduce the courier, the garbage truck you mentioned, the ARC protein.
The armored retrotransposon courier.
Right. Now, ARC, which stands for activity-regulated cytoskeleton-associated protein, is a master regulator of memory and synaptic plasticity. You absolutely need it to consolidate long-term memories. But structurally, it hides a very strange secret. It has retrotransposon origins.
Which means, and this absolutely blew my mind when I first learned it, millions of years ago, An ancient virus infected an ancestor of ours, and its viral DNA actually integrated into our genome.
It's one of the strangest quirks of evolution, honestly.
Yeah, and over deep evolutionary time, our bodies repurposed that viral code for our own use.
Precisely. And because of this viral ancestry, the ARC protein naturally forms what we call capsids. A capsid is essentially a 20-sided geometric protein shell.
Like a microscopic soccer ball.
Yeah, exactly. It is the exact same structure a real virus uses to protect its genetic material when it travels between hosts. ARC builds these hollow, virus-like shells naturally inside your neurons.
And the neuron apparently uses these armored shells to package things. The researchers found that ARC forms these capsids and then gets released from the dendrites, the receiving branches of the neuron.
But it doesn't just float out naked into the brain.
Right. It leaves inside a protective lipid bubble called an extracellular vesicle, or EV, and the cell uses a specific molecular docking protein called IRSP53 to physically pinch this bubble off from the cell membrane so it can float away.
It's a highly orchestrated export system.
But wait, I'm stuck on something here. If ARC is this beautiful essential protein designed for making memories, why on earth is it acting like a viral delivery service for toxic zombie tau waste?
That is a great question. It fundamentally comes down to cellular panic. You see, early in Alzheimer's disease, before the cells actually start dying, the neurons become highly agitated and hyperexcitable.
So they're firing constantly.
Constantly and erratically. Now, because ARC is a protein that is produced in response to cellular activity, I mean, that's how we build memories when we learn something new, this hyperexcitability causes a massive, unnatural spike in ARC production.
Ah, I see. So the cell is drowning in toxic tau, and suddenly it also has an overabundance of these empty ARC viral-like shells floating around just because it's firing so much.
Exactly. The cell is overwhelmed. It takes this normal memory building process and just hijacks it, turning it into a hyperactive transport system just to throw the toxic trash out and survive a little longer.
Wow. Okay, so we're about 5 minutes into this deep dive. Let's do a quick recap for you listening, just to make sure we're all on the same page.
Good idea.
So first, we have tau, a normal protein that misfolds into toxic, corrupting zombie clumps. Second, we have ARC, a memory protein with ancient viral origins that naturally builds protective armored shells. And third, ARC leaves the cell in lipid bubbles via a docking protein called IRSP53.
That's a perfect summary.
So it makes you wonder, if you were a cell filling up with toxic trash and you had a fleet of armored couriers just sitting there, wouldn't you try to use them to bag up the trash and throw it out? Let's see if that's actually what happens.
And that is exactly the hypothesis the researchers wanted to test. But, to prove that ARC is the specific courier bagging up the tau, they had to catch it in the act.
But how do you even do that?
Well, they started out in vitro, basically in petri dishes, using primary cortical neurons from rats. In normal wild-type rat neurons, they observed that tau was regularly being released in those protective lipid bubbles we mentioned.
But then they brought in the genetically modified rats, right?
Yes. They used ARC knockout neurons, meaning they completely deleted the gene responsible for making the ARC protein. These cells literally could not manufacture the garbage structs.
And what happened?
When they looked at the fluid around these modified cells, the release of tau had completely plummeted. It essentially stopped the export. Yeah, and they even went the other direction. They took specialized cells, neuro2A cells, and artificially overexpressed or forced them to overproduce ARC and that IRSP53 docking protein. When they did that, they saw a massive boost in the amount of tau being released.
So they can literally turn the faucet on and off in a dish. But biology in a dish is vastly different from biology in a complex living brain.
Exactly, which is why they moved to living mouse models for their in vivo evidence. They used a specific line of mice called the RTG4510 model, sometimes just called RTGWT.
And these mice are special because they are genetically engineered to express a human tau mutation, right?
Yes, the P301L mutation. This specific mutation causes severe tauopathy. The tau misfolds incredibly fast, forming those toxic tangles, giving the mice a rapid, severe version of Alzheimer's pathology.
So their brains are just filling up with toxic tau.
Right.
Then the researchers crossbred these sick mice with the mice that were missing the ARC gene, so you end up with an ARC knockout mouse.
Right, a mouse that produces massive amounts of toxic waste but has absolutely no ARC garbage trucks to move it.
And what do they find?
The findings here were monumental. When they isolated the tiny protective vesicles from the brains of these mice, they noticed something crucial. Deleting ARC did not reduce the total number of vesicles floating around.
Oh, really?
Yeah, the brain's general shipping and receiving department was working just fine. But the cargo was drastically different. The amount of tau packaged inside those vesicles was severely reduced.
Okay, but how do you physically prove that the ARC protein and the toxic tau are actually riding in the exact same vehicle? I mean, these things are microscopic. You can't just take a photo of them sitting together in a car.
You can't take a normal photo, no, but you can use an electron microscope. Specifically, immunogold electron microscopy.
Right. For you listening, imagine a tiny microscopic soap bubble. The researchers used a brilliant technique involving tiny, heavy gold beads to see what was inside.
Exactly. They took incredibly small gold beads, just 6 nanometers wide, and attached them to antibodies that specifically hunt down the ARC protein. Then they took slightly larger gold beads, 15 nanometers wide, and attached those to antibodies that hunt down tau.
And they use gold because it's so dense that the electron beam of the microscope can't pass through it, leaving solid black dots on the image, right?
That's right. You can clearly tell the small 6 nanometer dots from the large 15 nanometer dots. And under the microscope, they literally saw both sizes of golden beads huddled tightly together inside the exact same soap bubble.
So they were definitively co-packaged.
It's the ultimate smoking gun. But they knew that just proving they share a ride isn't enough. They had to prove this packaged tau was still dangerous, that it was still infectious and could seed new tangles.
So they used that biosensor assay.
Yes, the HEK293 biosensor assay. FRET relies on light emission. Basically, they engineered cells with tagged tau proteins. When a toxic zombie tau seed enters that cell, it forces the tagged proteins to clump together. When they get close enough, they emit a massive FRET light signal.
It's like a biological alarm system.
Exactly. And when they dropped the vesicles from the normal mice onto these cells, it triggered a massive FART signal. The packaged tau was highly infectious. But when they used vesicles from the ARC knockout mice, the ones without the garbage trucks, the signal was almost non-existent.
Wow. Okay, so we're at the 10-minute mark. Let's summarize this second segment really quick. First, ARC knockout cells release way less tau. Second, the total number of vesicles stays the same, but without ARC, they basically lack tau cargo.
Right.
Third, the gold bead microscopy definitively proves ARC and tau share the exact same vesicle. And finally, ARC packaged tau is highly infectious and seeds aggregation.
Perfect recap.
So for you listening, think about this. We've proved this beautifully in rodents, but mice aren't humans. What do you think happens when we look at the brains of actual human Alzheimer's patients? Let's dive into that.
This is where it gets incredibly real. Proving a mechanism in a genetically modified mouse is a great foundation, but does this actually happen in human disease? The researchers acquired postmortem brain tissue from human patients.
What part of the brain did they look at?
Specifically, Brodmann area 89 in the prefrontal cortex.
Okay, so an area heavily involved in complex executive function,
Yeah.
which gets severely impacted as Alzheimer's progresses.
Exactly. And they specifically looked at patients with brain stage 6 Alzheimer's, which is the severe end stage of the disease, and compared them to non-AD controls.
And what did they find in the human tissue?
They found the exact same mechanism. ARC and tau were consistently co-packaged in the human brain vesicles, just like in the mice.
That is wild.
It is. But crucially, the data revealed something even more striking. When they quantified it, there was a massive positive correlation in R value of 0.89.
Wow, R equals 0.89. That is incredibly high for biological data.
It's huge. There was this mathematically tight relationship between the amount of ARC in a human vesicle and the amount of specifically phosphorylated toxic tau.
So they're definitely riding in the same car in human brains too. But mechanically, how does ARC actually grab the tau? I mean, does it have molecular claws? Is there a pocket it slides into?
To figure that out, they turned to advanced AI and computational modeling. They used AlphaFold 3, which is an AI that predicts complex 3D protein structures.
Oh, right, the Google DeepMind AI.
Yes, and they combined that with Gromit's molecular dynamics modeling.
Gromit's Co.
Yeah, it's a software that simulates the physical atom-by-atom movements of proteins over time. They wanted to see exactly how these two objects collide and stick together in physical space.
And from what I understand, predicting how tau behaves is notoriously difficult because it's what scientists call an intrinsically disordered protein. Like it doesn't have a rigid 3D shape, it kind of just flops around like a wet noodle.
That's a very accurate analogy. It's highly flexible. But the AlphaFold and Gromit's Pixie simulations reveal that they form what's called a fuzzy dynamic complex.
A fuzzy complex.
Yeah. There are specific aggregation-prone parts of that tau noodle, specifically a hexapeptide motif called PHF6.
Which is an amino acid sequence, right? QIVY.
Yes, QIVY. When that specific segment gets near the arc shell, it forms a parallel beta sheet that aligns perfectly with a coiled coil region on the outside of the arc protein.
So for you listening, think of it like Velcro. It's not a single rigid lock and key. It's a bunch of weak, fuzzy, shifting contacts that stick together, which ironically is very similar to how normal tau sticks to the microtubule train tracks in the first place.
Exactly. It is a brilliant evolutionary reuse of an existing binding mechanism.
But I want to push back on something here.
Yeah.
Does ARC just grab any tau it bumps into? Like, does it prefer healthy tau, or does it actively seek out the sick, hyperphosphorylated tau?
They actually tested that directly using GST pull-down assays. Basically, they used ARC as bait to see what it would catch, and they found that ARC bound to the sick, phosphorylated tau with a significantly higher affinity than unphosphorylated, healthy tau.
So the garbage truck naturally prefers picking up the toxic waste.
Exactly.
Okay, let's do our 15-minute quick recap. First, human AD brains show ARC and toxic tau tightly linked in vesicles. Second, there's a massive correlation between ARC and phosphorylated tau. Third, they bind together like fuzzy Velcro via the PHF6 motif. And fourth, ARC prefers binding to sick tau.
Spot on again.
But this leads to a really difficult puzzle. If ARC is actively binding to sick tau and throwing it out of the cell, isn't that a good thing for that neuron? Let's look at what happens when you completely remove this garbage truck from a sick brain.
And this brings us to the core paradox of the entire paper, right? It's a double-edged sword. If releasing tau spreads the disease to the neighborhood but keeps the original cell clean, what is the net result?
Right, accumulation versus transmission.
Exactly. So to answer this, they looked at the CA1 region of the hippocampus in 4-month-old RTJ-ARC knockout mice. At 4 months, without the ARC courier to throw out the trash, the toxic tau accumulated heavily inside the individual neurons.
You typically couldn't get exported.
Right. And the result was brutal. It led to severe accelerated cell death.
How did they measure the cell death?
They used tunal staining, which is a chemical assay that specifically detects the fragmented, shattered DNA of dying cells. The 4-month-old mice without ARC had massive tunal staining and a significant loss of neuron-positive neurons, meaning the actual brain cells were dying off fast.
So early on, losing the garbage truck is disastrous. The house fills with trash and collapses. But that was at 4 months. What happened later?
By 8 months of age, the story completely changed. The total pathology, the widespread tangles, and the overall cell death were virtually identical between the mice with ARC and the mice without it.
So removing ARC didn't cure the disease, it just exacerbated the early toxicity.
Exactly. And by the way, they verified the same timeline paradox in female 3XTG AD mice at 12 months, just to be absolutely sure. Without the trucks, the first houses fall quickly, but the disease catches up eventually.
Okay, so we know removing ARC hurts the individual cell early on. But how did the researchers ultimately prove that ARC is the thing responsible for transmitting the disease to neighboring cells?
They designed what is arguably the most elegant experiment in the entire paper. They used an AAV tracking system.
An adeno-associated virus.
Yes, essentially a harmless engineered virus. They injected it right into the medial entorhinal cortex of the mice, which is where Alzheimer's pathology typically starts.
I want to break down the logic of this experiment because it is so clever. For you listening, the engineered virus forces the donor cell to manufacture a single protein chain. But embedded in the middle is a sequence that automatically snips itself in half.
Right, a self-cleaving peptide. When it snips, you get 2 separate proteins. One half is a bright green fluorescent protein, or GFP. The other half is the mutant toxic tau, the P301L we talked about.
So the donor cells glow green, ND. They have toxic tau. But the trick is the green glowing protein cannot spread from cell to cell. It stays put.
Exactly.
So the logic is, if the tau does spread out of the donor cell, you will find recipient cells nearby that are full of toxic tau, but have absolutely NO green glow.
It is a perfect visual tracking system, and the results were definitive. In the wild-type mice, they saw plenty of those recipient cells. The tau was spreading rapidly, leaving the green glow behind.
And in the ARC knockout mice.
In the ARC knockout mice, intracellular tau transmission was almost completely absent.
Oh, wow.
Without ARC, the spread practically stopped in its tracks.
That is incredible. Okay,
Yeah.
Quick final bullet recap. Without ARC, toxic tau builds up and kills the cell early on. But without ARC, the tau cannot easily spread to neighboring cells.
Right.
So for you listening, ask yourself this. ARC is taking the trash out of your house, but dumping it through the neighborhood. Which is worse? Losing one house early or infecting the whole block.
And that is the multi-billion-dollar question for the pharmaceutical industry right now. It completely reframes how we must design future Alzheimer's drugs.
Because right now, everyone is developing anti-tau antibodies, right?
Right. The idea is to flood the brain with antibodies that hunt down and clear out tau while it floats between cells. But if this research team is right, that strategy has a massive armor-plated blind spot.
Because the toxic infectious tau isn't just floating naked in the fluid.
Exactly. It is hiding inside a virus-like ARC shell, which is further wrapped in a lipid EV bubble. Those traditional antibodies might simply bounce off the outer layer. They might never reach the toxic payload inside.
So we might need entirely new drugs that slip inside the cell and prevent ARC from binding to tau in the first place.
Yes, maybe targeting that fuzzy Velcro PHF6 connection we discussed.
Which brings us full circle to that provocative thought we started with. The paper speculates heavily on the emerging role of viruses in Alzheimer's disease. Pathological tau hasn't just been shown to interact with this ancient viral ARC protein.
No, previous studies show it interacts directly with the capsid of HSV1, the herpes simplex virus, and that also promotes tau release.
And that connects directly to the data that shingles vaccines, which protect against a related herpes virus, measurably lower the risk of developing dementia.
It hints at a deeply intertwined relationship between viral biology and neurodegeneration.
It really leaves you with a haunting thought to mull over as we end this deep dive. The ARC protein is absolutely essential for you to learn and form memories. But because it evolved from an ancient virus, it naturally builds virus-like shells. What if the very protein that allows you to remember who you are is the exact same viral delivery system that Alzheimer's hijacks to steal your memories away? Keep diving deep, and we'll catch you next time.